Hi everyone. In this video, we will continue the next subtopics in chapter Immunity. 11.2 Development of Immunity. There are 3 learning outcomes in this subtopics. The first learning outcomes is to state the types of immune responses. Next, explain humoral and cell-mediated immune response against infection. And, the last learning outcomes is to explain the primary and secondary immune response. Types of immune response. There are 2 types of immune response, which is humoral immune response and cell-mediated immune response. Humoral immune response is the branch of adaptive immunity that involves the activation of B cells, and that leads to the production of antibodies, which defend against bacteria and viruses in body fluids. While the cell-mediated immune response is the branch of adaptive immunity that involves the activation of cytotoxic T cells, which defend against infected cells. When a foreign particles infects the body, it is taken out by macrophages via phagocytosis to form antigen-presenting cell, APC. APC is a cell that displays foreign antigens, as well as its own surface proteins. For example, dendritic cell, macrophages, and B cells. Major histocompatibility complex, MHC, is a host protein that functions in antigen presentation. Antigen that associated with T cell will use CD4 receptor that expressed by helper T cell, and CD8 receptor that expressed by cytotoxic T cell. Helper T cell is T cell that activate B cells and stimulate cytotoxic T cell production. Cytokines is signaling proteins that regulate interaction between cells in the immune system. For example, interleukin 1 IL-1 and interleukin 2 IL-2. B cell is the type of white blood cell that responsible for antibody-mediated immunity. When stimulated, B cells differentiate to become plasma cells that produce antibody. Cytotoxic T cell is the T cells that recognize and destroy cells infected with viruses or other intracellular pathogen. Helper T cells stimulate humoral and cell-mediated immune response. Number 1, show that the macrophage ingests a microbe or other foreign particle and breaks it into fragments. Foreign antigens. Number 2. MHC class II, self-protein. Bind to foreign antigens. Number 3. Foreign antigens will display on the cell surface. Number 4. Helper T cell recognize and bind to the MHC antigen complex. Caused antigen-presenting cell release interleukin 1 and diffuse to the helper T cell and stimulate it. Number 5. Activated helper T cell release interleukin 2. Interleukin 2 makes helper T cell itself grow and divide, producing memory cells and additional active helper T cells. Number 6. Interleukin 2 activate B cells and stimulating the humoral immune response. Number 7. Interleukin 2 activate cytotoxic T cells, stimulating the cell-mediated immune response, humoral immune response. Number 1. Once antigens are bound to B cell receptor in the B cell membrane, the B cell displays those antigens that bound to class II MHC molecule on the surface of its plasma membrane. Number 2. Activated helper T cell bind to the sensitized B cell. Activated helper T cell will release interleukin-2 that co-stimulate the sensitized B cell and trigger its activation. Number 3. The activated B cell then proliferates or divides methodically, producing memory B cells and plasma cells. Plasma cell secretes antibodies. Number 4. Memory B cells are long-lived cells that can give rise to effector cells if the same antigen is encountered later in life.
The activation phase begins when an invading bacteria is phagocytized, engulfed, by an antigen-presenting cell, APC. A lysosome containing digestive enzymes combines with the phagosome to process the antigens. The processed antigens combine with the MHC class II proteins and are presented on the surface of the APC. Helper T cells, CD4+, recognize the displayed antigen on the APC and bind to the MHC class II antigenic peptide complex. This binding triggers the APC to release the cytokine IL-1, which activates the helper T cell. The activated helper T cell releases the cytokine IL-2, which stimulates the T cell to proliferate. Thus, producing many helper T cells, each with a receptor specific for the original processed antigen. The effector phase begins when a B cell that exhibits on its surface an IgM receptor specific for the same antigen originally engulfed by the APC encounters and binds the antigen. The B cell engulfs the complex by receptor-mediated endocytosine. The phagosome containing the antigen fuses with a lysosome. The antigen is processed. The processed antigen binds MHC class II protein and is displayed on the surface of the B cell. Helper T cells now bind to the displayed antigen on the surface of the B cell, causing the helper T cell to release cytokines. The cytokines stimulate the B cell to divide and proliferate into identical B cell copies. The B cells differentiate, change and develop into antibody-producing plasma cells and memory cells. The plasma cells release antibodies with a specificity identical to that of the surface receptors on the parent B cell. These carry out the ultimate goal of fighting the foreign invaders. The released antibodies bind in a lock and key fashion to the antigen on the surface of the original bacterial invaders. This makes it easier for killer cells to attack and destroy the bacteria by phagocytosis, and the release of proteins causes the direct lysis of the bacteria. Simultaneously, a blood component known as complement is signaled to attack and puncture holes in the bacteria.
Cell-mediated immune response. Number 1. Helper T cell with its antigen receptor and accessory protein, CD4. binds with APC. APC releases interleukin 1 to activate helper T cell. Number 2. Activated helper T cell release interleukin 2. Helper T cell proliferate producing activated helper T cells and memory helper T cells. Activated helper T cell will release interleukin 2 and the cytotoxic T cell is activated. Number 3. Activated cytotoxic T cell undergoes mitotic division, producing active cytotoxic T cells and memory cytotoxic T cells. Number 4. Activated cytotoxic T cell migrates to the area of infection and eliminates cells that are infected by viruses or other intracellular pathogen. Cytotoxic T cell may release perforin to destroy the target cell's plasma membrane and form pores.
Okay, so in step 1, we're going to have a virus, and the virus invades a host cell. So here's the virus, and you can see here the virus, how it's invading the host cell. When the virus invades the host cell, the virus will take over the genetic machinery, right? They'll take over the cell, and the virus will produce viral proteins, and those viral proteins will become fragmented. Notice on this cell here, we have these self-antigens, which are MHC molecules. Those are already on the cell surface, just hanging out, waiting there. After that virus has invaded the cell and it's produced its proteins, and the proteins have been fragmented, now we can have your MHC molecule present the antigen on the surface of the cell. We're talking about cytotoxic T cells here that are going to have to bind to this viral antigen. So what class MHC molecule should be binding to this antigen and presenting it to the cytotoxic T cell? Hopefully you said class 1. Class 1 MHCs will present antigens to cytotoxic T cells. So the next step then is for the cytotoxic T cell to come in and bind to the antigen presenting cell. So here's your cytotoxic T cell, and it binds to the antigen. So what this slide does not show is the protein that's going to ensure or enhance the binding of the cytotoxic T cell to the virally invaded host cell. What protein is going to enhance this binding? Remember that your cell, we're dealing with cytotoxic T cells. So this protein that's enhancing the binding must be a CD8 protein. When the cytotoxic T cell, and this is something new, so it's very important to pay attention, when the cytotoxic T cell recognizes and binds to the class 1, okay, and then you get the CD8 enhancing the binding. At the same time that this happens, at the time of your cytotoxic T cell binding to the antigen, the cytotoxic T cell is going to receive a signal from the helper T cell. This signal is in the form of a chemical messenger called interleukin 2. Once we have this occurring, okay, and we'll talk more about this helper T and where this IL-2 thing came from here in just a moment, but let's talk about what the cytotoxic T cell does. We know that cytotoxic T cells, think about the name, cytotoxic. Cyto is cell, toxic, kill. So we want to kill the virally invaded cell. So how does that happen? Well, the cytotoxic T cell is going to release chemicals called perforins. And you already know that natural killer cells release perforins as well. And perforins will create a pore or a hole, right, in the membrane of the virally infected cell. So once that pore gets created, then we know this increases the permeability of the cell to water and ions, and the cell will lyse. Now, just to ensure that this cell is killed, something else happens. The cytotoxic T cell not only releases perforins, also releases a chemical called fragmentins. So this is a chemical that enters the cell via the pore that was created by the perforins. What the fragmentins do when they get into this cell is that they induce apoptosis, programmed cell death. They tell that cell to commit suicide. So here we have sort of a backup system, right? If the perforins that created the pore don't get the cell lysed very quickly, well, you can also rely on apoptosis or programmed cell death, and that cell will kill itself. So now let's put all of this information
Primary and secondary immune response. Primary immune response is the results from first exposure of B cell to an antigen. For primary immune response, low antibodies concentration produced at early stage has lag time to produce antibody. First antibody produced is IgM, and short time response. Secondary immune response is results from second exposure of B cell to the same antigen. For secondary immune response, IT will produce higher antibodies concentration, faster to produce antibody. First antibody produced is IgG, and longer lasting response.
When a B cell encounters an antigen that fits into its receptor, it starts to divide. This process is called clonal selection. Two types of B cells are produced: effector cells, which respond to antigens throughout the body, and memory cells, which persist in the body for a number of years and remember an antigen. It usually takes several days after exposure to an antigen for the B cells to form a clone of effector cells. About 7 days after the first exposure to an antigen, specific antibodies appear in the blood. If the same organism is exposed to an antigen that has already elicited a primary immune response, a secondary immune response is triggered. This response is much faster because the immune system remembers the antigen. The antibodies produced during the secondary immune response are often more effective against the antigen than those produced during the primary immune response.
Okay. That's all. Good luck for PSPM2.